What if the drug that could help someone with early onset GSD4 or adult onset GSD4 APBD is already sitting on a pharmacy shelf somewhere, approved, tested, and waiting? That's not wishful thinking. That's the actual premise behind a project we've launched with our research partner, COMBINEDBrain. Today we want to walk you through what this project is, what we hope it finds, and what we believe it could unlock even beyond that. But first, a number: 34,000. That's the estimated number of people worldwide who may be living with some form of GSD4 right now, most of them undiagnosed. Only about 370 have ever been formally recorded. Think of an iceberg. What's visible above the water barely hints at what lies beneath. We launched the hashtag #FindThe34,000 campaign because every one of those people deserves answers. And answers require research. Research requires data. And that's exactly what this project is designed to generate. GSD4, glycogen storage disease type 4, is caused by mutations on the GBE1 gene. That gene controls an enzyme responsible for how the body processes glycogen, the sugar stored in our cells for energy. When GBE1 doesn't work properly, abnormal glycogen, called polyglucosan, builds up and damages tissues including the liver, muscles, heart, and the peripheral and central nervous system. What makes GSD4 unusual is the breadth of its continuum, and that there is an overlap. In children, it can mean liver disease, heart problems, muscle weakness, and developmental delays. In adults, the same gene mutation can manifest as APBD, adult polyglucosan body disease, which is often misdiagnosed as MS or ALS, and can include progressive weakness, bladder dysfunction, and in some cognitive decline. Same gene, very different symptoms. And right now, no approved treatments exist for either. So where do we start when there's no cure? One powerful answer is drug repurposing. Instead of spending 15 years and over $1 billion developing a new drug from scratch. You ask, of the thousands of drugs already approved by the FDA, are any of them doing something in the body that could help our patients? This approach can shrink development timelines from decades to potentially just years, and the safety profile is already known. That's where COMBINEDBrain comes in. Through their biorepository platform, and with the help of a dedicated research partner, we are collecting nasal swab samples, simple, non-invasive, from approximately 20 to 30 patients across the GSD4 continuum, and including healthy controls. From those swabs, RNA, the blueprint that is used to make proteins in our body, is extracted and analyzed using a technique called RNA sequencing. While we only carry 2 copies of a particular gene, these genes can make hundreds or thousands of copies of RNA to make and store the correct amount of the proteins and enzymes necessary for our bodies to function. RNA sequencing tells us how much RNA is being made for each gene and allows us to compare how RNA levels are similar or different in patients with a GSD4 disease versus healthy individuals. COMBINEDBrain has partnered with Unravel Biosciences to conduct this RNA sequencing and use its unique platform to compare the RNA profile of GSD4 patients to the known mechanisms of thousands of existing drugs, searching for a biological match. The goal is to identify drugs whose effects on the body counter the effects of GBE1 mutations. Therapies that could push the biology back toward health. One of the things we are excited about is the ability to compare patients across the GSD4 continuum. Pediatric cases, adult APBD cases, and even pre-symptomatic individuals who carry the mutation but haven't yet shown symptoms. That kind of comparison could begin to tell us something important about why the same gene behaves so differently across a lifetime. The primary deliverable. A ranked list of already approved drug candidates to explore further through observational studies or possibly clinical trials. But the drug list is just the beginning. The RNA sequencing data generated by this project has a life far beyond drug repurposing, and this is the part we really want people to understand. This gene expression dataset is essentially a portrait of GSD IV disease at the molecular level. And it opens doors for our community that didn't exist before. It can help us understand what's happening in GBE1-related disease beyond glycogen metabolism. We know that glycogen builds up abnormally, but what else is disrupted in how cells signal each other, how inflammation is regulated, how energy is managed? This data can begin to answer that. It can attract researchers who want to study GSD IV, but haven't had access to patient data. Computational scientists, neurologists, metabolic disease specialists. This dataset becomes a resource they can work with, publish from, and build on. Finally, this data can lay the groundwork for biomarker discovery, measurable signals that could one day tell clinicians how a patient is progressing, or whether a treatment is actually working. That's something the GSD IV community currently does not have. We are at a turning point. The science is here. The platform exists. The samples are being collected. And the data we generate from this research study could ripple outward in ways that help not just the patients we know today, but over 34,000 others impacted around the world. To support this work or learn how to participate, visit apbdrf.org. Together, we can find what's already out there.